So I'd like to welcome everyone back. Thank you for your persistence and perseverance with this. You always wonder when a conference should end. I wonder if we ended it on Thursday night, would Thursday evening get quiet? But it's actually particularly good considering lots of people are traveling home. The reason why I think we need to have a closing session is that there's just been a lot discussed, and everybody's brain is thinking differently. And you know, we now have an exciting plan for the future, how this research conference is going to evolve. And with contributions from people like Liz and Randy and others moving forward, I think it sets the tone for a very exciting time for us. But also to do it in a way that we kind of work with and respect the other kind of more generic carotenoid conferences, that we can contribute to those special events also. So, as always, our esteemed colleague, Dr. Bernstein, is going to lead this session and try and maybe challenge some of us and maybe just present some comments or questions that will help us as organisers, but will help you as scientists and delegates to contribute back and prepare ourselves for the next event. So I'll pass it over to Paul. Thank you.
Okay. All right. Well, thank you very much. This has become a tradition. The third year in a row we've been doing this for the conference, where I've basically just kind of thought through all the things that I've learned, that I've seen at this conference, and thought of what's going to be happening at our next conference, whether 1 year from now, 2 years from now, or this one will be 3 years from now. We've all had a lot of new data presented to us. A lot of this, I'm very impressed at how much new data was here, both in the invited talks and in the posters here. And based on what I've seen here, I've just written down some comments and some questions that we can just discuss. And in the past, when we've done this before, I've broken it down just to one slide. This was the slide. You can read through this yourself up here. This was the homework slide from 2011, and you can decide how much. You'll see some of them are still the same questions we've been going at. Others have been a little more resolved, or we've moved on. And what I did, this is the 2011 one. The people who kind of gave the talks, you can see your initials up there, perhaps, of what you were supposed to be working on back then. This was 2011, and then this is the 2013 slide. And I was impressed by how much more information we had at this conference, probably even than the other two combined. So I couldn't fit it all on one slide. So this time I broke it down using John's divisions of the talk. There were about, I think, five different themes. Is that right? Or is it, I guess, six different themes here. Yeah, so there were 6 different themes. And so we're going to have a slide just on each one of these. Some of my comments, I'll read them, and we'll kind of just go through them together, especially the people who were the lecturers, or who did the posters, or the discussion leaders. I really want you to chime in. And I'm sorry if I've forgotten anyone. This was done after the dinner last night, so I did skip whatever event you had down and the dancing and whatever drinking you had afterwards. So I did have to do a little bit of work, and so you owe me a little bit for that. But so if I've missed things, I've tried to just stimulate the thought. I don't want to criticize anyone. I don't want to put anything, feature anything too heavily that shouldn't be. So we will start with the first one of the carotenoid source, and see if I can even read this. So this, and when we started here, I was struck by the fact that we still need to, with the early talks by Antonio and George Britton and some of the posters, we still need to expand our carotenoid diversity sources. And clearly there are over 600 carotenoids in nature. We know we've been focusing, or many of us have been focusing on lutein and zeaxanthin, and certainly there's a lot of remarkable specificity in the eye, but we do have to look at the more diversity of our sources and really think about that. So we can talk about that a little bit. I was also impressed from multiple independent directions. This really was the year of the chicken here. Much more egg work than we had seen before. So And we learned, I think, in some of the early talks, in the talk from Steve, it's Steve, right? Thurman?
David.
David Thurman, okay. That eggs are a surprisingly efficient source of carotenoids for getting into the blood, into the serum, and they can be manipulated. I put readily in quotation marks because after I heard that they had to deal with 8,000 eggs and laying chickens, I thought that was it's a challenge, and that's why you can see, even though it sounded like a good idea, I think in practice of trying to feed people for 2 months on these eggs was still a challenge, and we can talk about that in a second. And then, personally, from my aspect, I think biochemistry still counts, and I've put in, that's my first love in science is chemistry and biochemistry. I think that it's, the clinical studies are fantastic, but we have to remember the basic mechanisms. And I would say, compared to other instances of this meeting, there was probably a little less biochemistry. So I want to remind people you have to work with the cells in culture. You have to get out your HPLC and really look at the tissues and look at mechanisms, because mechanisms will direct our research and will really solve some of the problems that we have. So I'd like to open it up now to any of the people here, John or anyone else. Yes.
Great. So we're going to get the mic, yeah.
Yeah, and we'll have to watch the time. We only want to spend 5 or 10 minutes on each of these slides so we can cover everything.
So I'm going to be a little bit of a devil's advocate. You talk about carotenoid diversity, but are there other carotenoids in the brain and in the retina and in the eye that we know about?
Okay.
Like, why have diversity for these other rare carotenoids if we don't use them?
Okay. So I'll answer for the eye. For the eye, it's really lutein and zeaxanthin, and obviously mesozeaxanthin is the one that was, historically, I'm sure Bone and Landrum were very surprised when they found that isomer there. There are minor ones. If you talk to the Japanese groups, they are very interested in astaxanthin. They think that some of these carotenoids may get in. We know things like canthaxanthin can crystallize in the eye, so there can be toxic effects. So others may be useful, and they may be useful for pharmacology. That's the thing. So just because they aren't naturally there, we at least want to look into them. So go ahead. And then
isn't astaxanthin mostly localized in the sclera, though? It's not really. Is that true or not? I'm not sure. I mean, there are experts here.
Astaxanthin doesn't get in very well in my. We rarely
doesn't get
see
into the neural structures.
Yeah.
Is that correct? Okay.
Yeah. Do you want to say something, John? Go ahead.
Well, I'm with you on the biochemistry. Back to the diversity, though, I'm not so sure that we need a diversity of carotenoids. So much as what we heard is there are diversity of emerging sources of the existing carotenoids. And I really would like to know if some of these food sources might deliver as parts of the diet more efficiently the carotenoids that they bear into the blood. And so I think we're seeing emerge some new food sources. And how good are these as food sources for these carotenoids might be a good question. And I remember as a kid, kiwis were a very unusual fruit, but they're all over the place now, and now we're seeing other fruits come into the market. But it's again the same thing. Should we be pushing to see some of these be commercialized, and is there real value in that aside from just their uniqueness?
Right, and also obviously improved sources too, as we were seeing with plant breeding or genetically modified. Yeah, Liz.
Yeah, I want to pick up on something John was talking about, is how good are these new sources in terms of uptake. And I am working on a project right now that's looking at bioavailability and how we can incorporate that into our food frequency questionnaires to fine-tune that tool to be a better marker of status. And so that's something I'd like to see considered is bioavailability. We know it's better for eggs, it seems to be better for fruit compared to vegetables, better for cooked versus uncooked. So to see that consideration when we assess these intakes, I think would be important.
Yes.
Just to follow on from those last two comments, with the zeaxanthin biofortified sweet corn, with the coming onto the market, the hybrids have been finished and they're just going through expressions of interest now for commercialisation. From a bioavailability perspective, just starting with the bioaccessibility, it's been really interesting because we were trying to work out where we knew that the zeaxanthin and lutein weren't, as the levels went up, weren't forming crystals, and they weren't just hanging around. And we were thinking, well, where the heck are they? And we're thinking at the moment that the zeaxanthin's actually being protein bound in these zein helix storage proteins. So how that goes with bioavailability is still something we've got to work out.
One or two more comments, then we'll move on. Yes.
In terms of bioaccessibility, I wonder if the comparison between the two pharmaceutical preparations, which I showed in the lecture on the eggs, because there you have two preparations of similar composition, but one has both have different fat sources, soya bean and sunflower oil. Yet there was quite a big difference in the bioaccessibility or bioavailability of those two sources. Now, if anything, the Macular should have been more effective. It was much smaller particle size, according to Carlos. So there should have been improvements. It should have been much better. But in fact, it appeared to be poorer of the two. So that might give us something to look more closely at. Those two preparations might help us in looking at bioaccessibility.
Next one?
Yeah, I think so.
Okay. So we're talking about predictors and functions. So the comments that I had is we still have to optimize and improve our methods to assess macular pigment. This has been ongoing. But I was particularly impressed that there are new methods still coming out that we are not done with this. So we may argue what's the gold standard, what's the best way, but I think that lots of new ways bring us new information. And I was particularly impressed on the FLIO work, and clearly I know that she had to leave already, so she's not here yet. But that work coming out of Germany on using time-resolved fluorescence spectroscopy is really very innovative. It's taking advantage of the Heidelberg Spectralis platform, which is really a very versatile platform to do measurements. And going back to my work, as you know, Raman spectroscopy is something that I've always liked, and I'd love to convince Heidelberg to add Raman to its, because it should be done. If you can do time-resolved fluorescence on carotenoids, which is really, you're getting to photon counting, and if that works, Raman, I think, should be easier. And then the polarization, using looking at Heidinger's brushes as a potential low-cost screening device, again, I thought is very innovative, and we'll see where that goes. So we can talk a little bit more. We don't want to go into this too detail. We could go on this, obviously, for days, probably. And then the other thing from this particular group was just that genetics, stress, and inflammation interaction with carotenoid status need to be explored further. There was a lot of good preliminary data, I would say. Nothing that I would categorize as totally definitive. There's still a lot of work to be done, but we can discuss where we go from here. So I'll open it to the floor.
Yeah, I'll start if that's okay, Paul. I think one of the things that still amazes me is how unpredictable macular pigment is. From something that we know is entirely of dietary origin. And we see with Julie's studies all the work done looking at predictors and even advancing looking at genetics as contributing predictors. We still have so much of this pigment in the eye that's not explained. And that, to me, suggests that there's something obvious that we're probably missing. And to add to that, it's not just a predictor of the amount of pigment we have at a given time. If we are trying to optimize this pigment, whether it's by sweet corn or diet or by taking supplements, I think understanding predictors of response to supplements, and that obviously starts the point David is making in terms of how it's encapsulated, how it's provided. There's just so much work I think needs to be done in that, because if the opportunity of the exercise here is to enrich pigment in some way, I think understanding it more. My own PhD thesis was determinants of macular pigment in healthy subjects. And we looked at in that study, I looked at over 1,000 people, and I still had about 78%, 75% unexplained variance in something that we know is dietary of origin. So I think that's a question that we should maybe bring back into to try and work with Julie and help in this research question.
Yeah, I think it's complex, and we can only understand them to the extent that we can measure them. So we keep breaking onto new horizons that suggest other things that might matter, like how will early life exposure to carotenoids affect, for example, the development of the fovea and ability lifelong to accumulate macular carotenoids. Haven't figured that in. And also when we look at diet, we're often looking at a snapshot of, at best a year. And so we don't know, how other things that modify carotenoids have changed in the diet in the years prior, or even in the weeks prior to when measurements are made. So there's some things we're not going to get a handle on. But I think in the foreseeable future, we might be able to understand quite a bit more about the genetics. The studies that we did were done with candidate genes for proteins that so far scientific studies have suggested are important. But I think we could do more by looking genome-wide. So certainly that's on my list to do things. And I think if we can then get bigger samples to look genome-wide, we might be able to develop a few boxes of people who might need more and less levels of carotenoids, or for whom benefits might be greater or less, and safety might be greater or less for certain levels. So I think the short-term target would be roughly understanding how differences across people might impact requirement needs.
Thank you.
Richard? Yeah.
One thing that I think is really important was brought out in one of the posters, and we've certainly seen it at other conferences, and that's the problem that the different measurement techniques often give extremely large differences in the measured macular pigment. And I think our goal should be that we can use any instrument we want, we can swap instruments partway through a study, and we should always be measuring the same thing. That would be a wonderful situation. But if you compare results from two different instruments and you get sometimes such a huge difference, then when you're trying to, say, relate macular pigment to something else, like serum lutein, for example,
That's the problem.
then you start to wonder, well, can I really rely on this data? Because maybe the technique that I'm using isn't really measuring it as accurately as it should be. So I think that would be a goal, to explore why the different techniques give the different values, and is there some sort of legitimate correction factor that could be incorporated.
But you do have to remember there are different assumptions. Should we be measuring the integrated area versus the one thing that I've been struck now that I use the spectralis is I know that in flicker photometry, often we concentrate on measuring the macular pigment at 0.5 degrees. Well, when I look at that slope there, you're on an extremely steep part of the slope there. So tiny measurement errors are going to give you different macular pigments. And so we have to look at, now that we have new information, new ways of looking at data, should we know that looking at distributions might be important, and we have to decide, what does it mean to be on the slope versus an integrated area, or should we be looking at the width at half maximum? There are all sorts of ways to be looking at the data that people haven't even started to look at yet. Yes?
You recall at the end of Billy's talk, he mentioned that he'd be startled to find more than about a 0.3 correlation factor for any of these things. And of course, Julie's correlations are suggest maybe 3% for this, 3% for that, and they add up. But, for the macular pigment, it appears the big vehicle for getting the carotenoid to the eye is the HDL. At least this is one of the predominant theories we hear repeated again and again. And so HDL ought to be a big actor, maybe even a high correlation actor. And there are genetic variants. And I was reading a paper not long ago where there's a suppressor that stops LDL from building up in the blood by causing it to be metabolized out in the liver. So there's some very interesting human variants in the lipoproteins that might produce candidates that would give us a handle on, in one or two people, learning about mechanisms of transport where we really understand some genetics.
Is it not true that maybe the carotenoids are preferably carried on HDL, but they're also carried on LDL, right? And we've looked at that. We've looked at that in Waterford, and we really didn't find anything that was strongly suggestive.
Okay, two more comments, I think. Okay, Lisa, you can go.
Okay, to your point, John, we published a paper a few years back where we actually gave a statin intervention, and we gave two of them. We gave atorvastatin, which drops LDLs and doesn't really do a whole lot to HDLs, and then we gave rosuvastatin, which drops LDLs and raises HDLs, and we saw a significant drop in macular pigment, not centrally, but not, getting sort of farther out with eccentricity when we gave the atorvastatin and not the rosuva. So even, that gives a little bit of insight into your question. When you dropped HDLs and dropped LDLs and didn't do anything to HDLs, we saw a decrease in carotenoids in the retina. When we boosted HDLs while dropping LDLs, we didn't see that same loss. So that may give some insight into your question, and I think looking at the transporters of these carotenoids is actually a really good idea. There's a little bit of empirical data out there that would suggest that this is so.
Yeah, thank you very much. I just wanted to make a comment, really coming back onto the HDL, but moving a little bit beyond that as well, to ask whether or not we could consider the stress and inflammation to be linked by metabolism, because many of the genes which I've seen being affected or been associated with carotenoid intake have also been associated with metabolism, or are actually key parts of metabolic pathways. And we've also heard about switching metabolic phenotype or switching macrophage phenotype as potentially one of the outcomes. And that can also be driven by changing in the cellular metabolism from more glycolytic to an oxidative phosphorylation pathway. And I think that's an area that is underexplored in this field, and I think that might be worth adding to the list.
Okay. We'll take one more comment.
Yeah,
and we'll
just to keep in mind that not all HDL is alike, unlike LDL, that we have all these different proteins. And when we've looked at that with Ernie Schaefer's lab and looked at alpha A1, some blah, 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 that's a pretty good marker for heart disease. We did find that change in this protein, or amount of this protein, was related to the amount of macular pigmentation with a lutein supplementation. So that's just something else to keep in mind. And when you talk about HDL, it's really a heterogeneous group of molecules.
Can I make one final point, Paul?
Sure.
Yeah. I think we have to go back to the first point made, and Mikey asked, can we try and do something that, that we can try and standardize our measurements? Lisa's been trying to suggest this for years, but we all go away and don't do it. Can we kind of charge someone with the responsibility of actually doing this and allowing us get the value from all the many studies?
So I actually started this project.
Yay.
And I have a big table in my inbox that has all of your studies in it. And I need a little bit of data. So I was going to actually ask you if I could have a couple minutes at the very end to say, look out, there will be emails coming from me, and we could form a work group and publish under the name of the work group instead of any one individual getting a ton of credit for this, because this really has to be a group effort. But I started this project a while ago, and I've got the table ready to go. I just have a couple of questions for some of you regarding some standard deviations and things like that you may have had in the past. So thanks for that prompt, and stay tuned for an email, folks who have done any sort of supplementation work in the past.
Okay.
Yeah, thank you, Lisa.
All right, next slide. Alzheimer's disease and cognitive function. That was definitely a new and expanding area here. We heard some talks that the brain, like the eye, is definitely a carotenoid target organ. There is specific uptake. There's intriguing new data that was presented that point to a relevant role in promoting cognition in normal and Alzheimer's disease patients. We can talk a little bit more about, this I'd still consider somewhat early stage, but it's an interesting pathway. And clearly we need to do larger, more definitive studies need to be done with improved assessments, and Lisa was talking about that in her talk. And then Martha presented at least some very early but intriguing data. It's going to be great data when it comes out on her monkey studies that are just like the monkey studies that were done 10 years ago on metabolism in the retina. This is going to provide a wealth of new information on early stage metabolism in the brain. So I'll open it up to where we should be going from here. Yes.
Hi, I think I just modify that to promoting cognition in normal and possibly Alzheimer's disease patients. I think it's just suggestive to my mind to suggest that maybe Alzheimer's is the target. Certainly mild cognitive impairment is, and mild cognitive impairment is sometimes represented as very early stage Alzheimer's disease. But to be honest, mild cognitive impairment is much more heterogeneous than that. A lot of people have mild cognitive impairment and it has nothing to do with Alzheimer's disease. So I think there are reasons to continue to see Alzheimer's as a possible target, but the large definitive studies we need to do first need to be looking at normal into that very early stage MCI, mild cognitive impairment group, and I think that's where the definitive large studies need to be targeted.
This was written late last night.
And from a clinical trial point of view, that's something Waterford is committed to trying to add knowledge to.
Yes,
one thing I
Martha's
just
next.
wanted to add is I feel like we know so much less about the mechanisms of xanthophyll actions in the brain, and that we need mechanistic studies, in vitro studies, whatever, looking at, what's being modulated in the brain. I think some of Liz's stuff is starting to address that with the isoprostanes, neuroprostanes, but there's just so many other possible mechanisms that they've been suggested, they've been speculated about, but we just know so little.
Okay, Randy or Matt, either one. Randy's next.
So one question I had about Neil and Liz's data is it seemed to me that Neil's data showed that xanthophyll accumulation was preferential to gray matter, but then I thought that your data showed that it was preferential to white matter, like myelin. So was that just an inconsistency? I couldn't quite figure that out. Didn't it preferentially go into myelin though? Did that seem like the part of the?
Well, that's the membrane we were looking at.
Yeah.
The myelin membrane.
So within gray matter?
In the striatum.
So I'm a little perplexed about that. I've been contemplating. It makes sense to me that it would go into the more lipid-heavy areas of the brain, which is white matter, not gray matter. But I thought Neil really suggested gray matter. I guess Neil's not here.
Yeah. If I could just, the question is whether they go. I mean, all areas are lipid-rich, but more polyunsaturated lipid-rich, or more saturated lipid-rich, like myelin, or monounsaturated and saturated lipid-rich, like myelin. That's Okay. But I think Liz's studies are starting to address.
Yeah, and
at least where is it?
Yeah, where is it? That's the first question. Where is it? And these were preliminary data. We're going to be adding to it to see if that still holds up.
Matt?
Yeah, well, in terms of whether it has an effect on myelination, which is a whole other level.
Matt?
So my viewpoint of that distribution in brain is that whole data set smacks of me to non-antioxidant activity. And as a build, just before lunch, we began to touch on some stem cell work. So I just wanted to let you guys know that in 3 years' time, we are poking around in stem cells in an effort to understand in a more purified system mechanisms of action for lutein and zeaxanthin. And in 3 years' time, we should have some very nice data to share with you guys.
And I think one charge that we'll take away, we have a lot of behavioral data here. You know, it's sort of the end of the spectrum of cognition, the end result of cognitive activity. But one thing that we are, as you saw in the talk, starting to do now is look through our neuroimaging data. So we have hundreds of subjects who have either gone through EEG, functional MRI. They're human subjects who will be getting DTI. And you know, the same measures that you were looking at, Martha, our humans are all getting. And these are young, healthy subjects and older adults. So one charge that we will take back is to really dig into our neuroimaging data and see if we can take a couple of steps back in level of analysis and see what we might find in terms of functional activity associated with xanthophyll levels.
Go ahead, John.
The brain is way too complex for us to understand, clearly. We're going to learn snippets here and there, and we're going to get some kind of model for it, which is very simplified. Single neuron function might be a place really where we should think to come back to. I mean, back to the Galvani sort of experiment. You know, you get an action potential from a single neuron, and how does lutein and zeaxanthin alter the behavior of a single neuron to a variety of stimulus? You know, if we've got a nerve function here, it ought to manifest itself at the single neuron level, at least in some fashion. And the question is what model for that single neuron form can we get? It's clearly we're not gonna find it easy to do human single neuron studies, but maybe there is a way to do this with primates or other sources. I mean, the neurobiologists always use these lobster neurons, right? But that's not going to work for us.
So I can build on that comment, John. I would expect in 3 years' time that we will have data like that generated on human stem cells that are encouraged to differentiate into single neurons. The other comment I wanted to add was that I hope in 3 years' time that we will have additional data to present from the CNLM, our venture at the University of Illinois, which were not discussed at this meeting. We are busy doing imaging studies in infants receiving lutein, and I hope we'll have some of those data to share then.
Yeah, just a comment on your top line there about target organs. So I could argue that lutein and zeaxanthin might build up in the brain because the other carotenoids are getting there and are being utilized for something or being metabolized. So just because something's there doesn't mean it has a function. So it could be, and I don't believe this, but it could be lutein and zeaxanthin is doing nothing and staying there because it's not being metabolized. You know, I don't believe that, but it is a possibility.
Martha? Oh, I guess you're up. You're next.
Hi. I wonder if anybody's looking at astrocytes. And we're focusing very much on neurons here, but they're actually supported very extensively by astrocytes. And there's some interesting work coming out about hypometabolism in astrocytes. And one of my colleagues is doing some nice work in that area. But I've no idea whether we know anything at all about macular pigments in astrocytes.
And the astrocytes are involved in delivery of DHA to neurons, so there's another parallel. But I wanted to say in response to John, it may not be about the individual neurons, but about their connections, you know, where the action is. And in response to John, that one way of conceptualizing what he's saying is that there's, you know, alterations in primates in the carotenoid metabolizing enzymes that may have been an evolutionary adaptation for the fovea, and that just, you know, it sort of happened to leak over into the brain. You know, I mean, the absence of breakdown of the carotenoids, it's sort of a brain epiphenomenon. I mean, I don't believe that either, but that's one way of looking at it.
I think it's certainly remarkable that you do see whenever there's a specificity, you know, even this isn't a lycopene conference, but people should be, have looked at it in relation to prostate cancer. So, it's less interesting when your tissue has carotenoids that are similar to whatever's in your bloodstream and in your diet, and that would be fat, maybe skin. Some of those show little specificity, but they're carotenoid containing. But the specific ones, I think, at least as a biochemist, those are the interesting sites to be going to, and the brain's at least showing that as a possibility. Okay, I think, are we ready to move on?
Yeah, and I think,
I'd just like to say one more thing about astrocytes. Lisa and I, when we first sat down and talked about what mechanisms may be behind the findings that we were seeing, astrocytes came up first, and I think astrocytes are a great target, one because of their oxidation purposes. So for instance, the astrocyte shuttle model is the theory behind the basis of fMRI, which is the bold signal, the oxygen being delivered there and it changes the signal. That's how you get the signal of fMRI. So I think astrocytes, personally, I think that's a great place to start.
All right, we'll go to the next one. Clinical studies. And first, we obviously heard a lot about the first phase CREST trial. And I mentioned this before, and I want to emphasize it again. It shows the value of doing a well-designed, executed clinical trial that does provide very good answers. And I think it's very good to learn from the fact John's been in this field for a long time. He did a lot of preliminary data, but based on his preliminary data, which could be criticized as being exploratory or not as well done as it should have been or not adequately placebo-controlled, it really led to a design of a very good clinical trial. That's how he picked his endpoint, and he found an endpoint. His primary endpoint was met, which is really not that common in clinical trials to do that. And so that's something to be said. And so we await the other results that will be coming. I do have a question for John that I'll open up on that study, but we'll finish off with the other ones. Is that we also learned that formulation composition need to be optimized. This was a trial that looked at the 3 carotenoids together versus placebo. That's a good thing to do. It gives us an answer, but is that the optimal formulation? Well, that means we have to do yet other trials. And so that's something that has to be on our to-do list. But these trials are expensive to do it right. But now that it's been done right one way, it can guide future trials. We learned that glaucoma could be the next frontier, and we can see if we want to discuss that anymore. And we all need to continue to initiate, execute, and support future studies. And I'd like to open up the discussion with a question to John. You reached your clinical endpoint of improved contrast sensitivity, but I don't think I didn't get an impression from this. What does that mean to us? What is an improvement on a 6-cycle degree?
On average, in the size letter, that for the cycles per degree, which is 6, clinically it means on average 3 letters on a vision chart. That's what it means clinically. So how you extend that to what it means in the real world in terms of outside, driving, looking up in the sky, Randy would be better positioned to maybe help me answer that. How that extends, how something measured on a vision chart extends to real life, Randy. Would you have a comment on that?
Sure, so the answer is
terribly. All right, there you go.
Yeah, I mean, Cynthia Owsley, who does a lot of driving studies, will try to correlate contrast sensitivity function measured in the classic ways to things like real driving performance, et cetera, and they barely correlate at all. And most of the reason is because outdoors, there's just different optical factors that one deals with. And in fact, one thing I've always argued is that, we didn't evolve the mechanisms to accumulate macular pigment indoors or with artificial lighting, or, so to the extent one can match the ecological conditions under which we evolved, you're more likely to see a functional effect of the pigments, I think.
Thank you.
So I think I have an answer to that that's unfortunately not a very good one for us, okay? If you take a camera and you take a photograph, you have the subject in focus. There's a certain depth of field in that focus. You can get a little bit behind, a little bit in front. anything that's outside of that narrow plane is out of focus. Now, when you and I look, we're looking through a lens, and it has the same qualities as a camera, but we don't see that. We don't see it, half the room is out of focus. And the reason is we've got a pretty large processor sitting behind that camera doing our integration, and our eyes jittering around and doing things. And so when you're making a glare measurement in the clinic, it's like taking a snapshot with the camera. And when you put us out driving a car, when we go out of doors, we're using a much more integrative visual processing system at full tilt. And so you've got a lot more going on here that's being covered over by a very complex mechanism that's evolved over, literally millions of years to handle these kinds of signals. So it's not surprising, in fact.
But yet, typically, the world measures vision in this way, even more crudely than that.
So the bottom line is a photograph is not a very good picture of reality for what we see. We may like the photograph, but it doesn't necessarily give it. So we need another measure other than that. That is what I'm saying. And I don't have a quick answer for what it might be, but it's in a way not surprising that when you measure at a pinpoints level that it doesn't correlate out to light.
And did you see the three letters on visual acuity? Could you also?
No,
see
there was no significant change in visual acuity.
Didn't see that. Okay.
No, which is consistent with previous studies.
Yeah, Mike.
Well, I mean, my world is clinical trials. And what I found actually in the carotenoid field in terms of clinical trials is that you don't do it the same way that we do it, like for drug trials. One thing that like I was in the AREDS-2 study. One of my comments when I was in the AREDS-2 study was that, why don't we have a dose response? How do we know that the amount of DHA that we use is correct, right? I mean, how do we come up with 150 milligrams of DHA? And also, same thing with lutein. Same thing with, we have to, when we do a phase 1, phase 2 trial, we have either a dose response where we ramp up to see biological effect and biomarkers. Most of the studies that are done in the carotenoid field don't do that. They just come up with the dose and then they go with it. Now, I mean, why? Understandable. It's because money. That's what you got. It's because it's money, et cetera. Right. But to ramp up the clinical trial side, you sort of have to work on that. The other thing is that I like the way the CREST study was designed because we have a problem with visual acuity. That's a big discussion in the clinical trial field because it's a subjective situation. Contrast sensitivity, threshold, light adaptation, these things, these dark adaptations, we're trying to incorporate them into clinical trials so that we have something that's objectified and we can quantify. Right now, contrast sensitivity is one of the more objective measures. Visual acuity sucks. It always has. And that's why we can't really do studies with visual acuity, but we're forced to because the FDA in the United States tells us we have to. Comment.
I think I've heard Randy say before that, it's not so much that visual acuity sucks, but it's just one part of a measurement that... It's like 5%. Yeah, but... overall vision.
Well, and I would also argue that it's not just money preventing us from doing clinical trials like that. What is your placebo, really? I mean, so it is the case in our clinical trial, which is a double-masked, placebo-controlled trial that controlled for everything under the sun that we could think of to control. And I still don't know what my placebo group is for lutein. I don't have a group of people who has never encountered a vegetable. I mean, this is not, I don't think that we should aspire to do FDA-style clinical trials if we can help it.
Else? We can move on. Okay, so now we're to the last two, which of course were just this morning. So these are the questions that I anticipated. So the ones that I've put here is, how do we go beyond AREDS-2? At least in clinical practice as a retina specialist, that's what they're looking at, is what is, AREDS-2 is guiding a lot of the supplementation, but we're the people here that should be at least thinking about what would we like to do if we had an AREDS-3. There's no money for an AREDS-3, of course, but we could at least talk about that. We also can talk, this is related to the earlier talks. Is there a link between macular degeneration and brain function? That's just an open question that I wanted to pose. And can we better understand the macular pigment's protective functions at a molecular level? These are just a few questions to open up, but we had all the talks just this morning. So, any comments?
Well, Harrod's tree, I mean, primary outcome measure, can we ever really map AMD as an outcome measure in that way? And particularly when you look at the variance in terms of subjects that were recruited into ARIDS, and when you look at, as you said today, Paul, you know, a lot of the subjects recruited into ARIDS already had way beyond, you know, what we see as kind of average macular pigment, etc. I think there were so many issues in terms of the design of that trial, but yet in terms of ophthalmology and what maybe they do now in clinic, it is guided by that big famous ARID study, which has taught us a lot. So I suppose the question is do we think there's a need for an ARIDS tree? Is it something as a community we should try and see, could we get all the funding agencies together to help us do something that's useful? If you had a clean page, what would it be? What would the next big clinical trial? For me, CREST was my attempt at a carotenoid-focused ARIDS, albeit with tiny numbers in comparison, but powered enough to look at visual function. So that's kind of a comment, but
I don't think we can do an ARIDS 3. We certainly can't do it with NEI funding in this era. But we can make use of existing data, again, when we get a large database. So there's something called Mendelian Randomization. You can look at incident eye endpoints relative to exposure to carotenoids, whether supplements or diet, in people who have certain genotypes and don't, or in people that have certain phenotypes that promote the expression of genotypes that are carotenoid promoting. So you can't randomize people and follow them prospectively, but you can use gene and metabolomic data, which can group people into two different comparison groups that might often. I have to say that I'm not well versed on the technique of Mendelian randomization, but as if it were a clinical trial. So I think that we can explore how to use existing data in more informative ways.
Tunde?
So I think we can do a lot with existing data. Sorry to repeat, but what I wanted to say really is that there are lots of cohorts out there, and especially now in Europe, that we unearthed a lot of them who actually measured, or sorry, they haven't, who have some data on their macular pigment, and they didn't even know if it would be useful or not. People are collecting a lot of things. So in the European Eye Epidemiology group that now has about 24, 27 big, quite large European studies involved, we could look at who has taken those images, because a lot of the times they take the images, but they don't have the expertise to analyze. So this group could facilitate sort of analysis of some of the macular pigment images were taken. And also, there are quite a number of groups that are either just in the middle of conducting follow-ups, 5, 10, 15-year follow-up, or the Tromsø study actually has been running since 1974. And they have just started their Tromsø 7, which is their new follow-up. So adding, if we could facilitate it, but a lot of these studies are very, very, not just cash poor, but resource poor in a way of not knowing the right people. So it took us, several years of not being able to get any shape or form something that we could measure macular pigment in Tromsø, because it's not a massively interesting big market. So I think there are lots and lots of ways of working together with existing groups that would then, just 6 months of a spectralis there, we could have had 2,000 patients in. with no problems. So I think this kind of stuff would be extremely valuable, and then we can go back, because if they have follow-up, especially those who have had food frequency questionnaires from 1974 coming up till now, even if food frequency questionnaires and things are not necessarily the best way of measuring everything, but some indication would fall out with a 30-odd year follow-up.
Yeah, I mean, the danger is this is the type of suggestion I love at these discussions, but what happens then is that people kind of go back to their own habitat, and we don't get it done. I wonder, should we be a little bit more decisive and say, look, say to me as the chair, can we get funds to achieve, maybe we should, after this meeting, all go away and think about the most important thing we need to do collectively.
Well, I mean, to come back to my suggestion, if we can look at, because I'm sort of co-founded the European Epidemiology work group. We've got a yearly meeting of all stuff, so we always the last couple of days of June. We can. I would love to think about with people like you. I don't really know what to do when they put 2,500, 3,000 macular pigment measurements in front of me. This is the right group of hoping to help.
Julie Mars would.
I know, but it's the question is, I've got the graders, I've got the I can get the images, but I don't really have the expertise to know what on earth I should be looking for necessarily on them. Once we know, giving it to a grader, those of you who worked with Reading Center, we're not as expensive as Wisconsin. You can then go ahead, and then you get historical data. So not retrospective cohort, but a historical prospective cohort you can put in place, which in terms of their value, they're very, very different.
So,
Thank you.
So I'm hearing some interesting things that make me think, as president of the International Carotenoid Society, that these are the kinds of things that a society can mobilize and accomplish that a few individuals cannot. I think many of us here are members of the International Carotenoid Society. Those who are not, I'd really dearly like to suggest you join. But we can, as a society, provide resource for communication. For instance, if you want to archive data through the International Carotenoid Society through our website, that's something we could facilitate. We can't fund at our size studies or that sort of thing at this moment, but we could, if a small set of groups wanted to nucleate around our society and through our society, do that kind of thing. And so if someone's got ideas in that direction, certainly the society would be open to the ideas and consider how it could help.
Yeah, and I'd also be willing to look at how this Macular Carotenoids conference community, how I can maybe establish something to, even if it's funds that's required to help maybe Julie correspond with Tunde and whatever piece of work, I'd be happy. Julie, if it's something that you feel we can do, let me know, and I'll speak to the various organizations that have made this possible, because...
Mike?
Yeah, I think building off of Tunde's
Yeah.
structure already, say, for AMD classification, so you've done some of that work with the European group. So I worked with Carolyn Popper specifically on some harmonization, but there's people like Cecilia Quartz who brought in. So you might be able to, we could use your structure instead of just inventing a new one and incorporate, let's say, macular pigment as an independent predictor and some of that. And then pull in the
Another model is something called the DRCR in the United States. The DRCR is a conglomeration of retina doctors who volunteer for trials that are designed by a group. And it's NIH funded, but it's a lot less than like doing an ARED study, because we pretty much volunteer our data. So like you could get a group of doctors here who see patients who have cohorts. And then the International Carotenoid Society or this body could actually be the organizers. It takes a lot less resources. It's more about administration and then recruiting doctors to do that kind of analysis. I mean, that would make a lot of sense because you could play a leadership role as the Carotenoid Society in forming this type of network.
Okay, we're going to take just one or two more questions so we can get to the last section. Torbert?
So, at your international conference, if this meeting is going to be in 3 years, you might what? No, your meeting. No, this meeting will be 3 years from now. So right. So maybe you could have a workshop on some of these things that involve getting people together for half a day. I don't know how your meeting is organized, but I do want to go to the next one. So you don't lose some momentum. So that's something to think about. Yeah, with the Gordon conference as well.
Yeah.
Yeah, so you don't lose momentum. Like there's all this cognitive stuff is so exciting.
Okay, last comment on this section here.
One area which I think is exciting we haven't mentioned so far, and that is Tos Berenschot's observations on the effects of lutein on complement factors. Complement factors are also raised in Alzheimer's disease, and nobody's looked at the effect of lutein in reducing complement factors in the serum of Alzheimer's patients, as far as I know. There's a lot of work looking at these abnormal complement factors in the pathology of Alzheimer's brains, but this would link both macular disease, Alzheimer's disease, our interest in lutein, and the inflammatory process, which presumably it is countering.
All right, thank you. We're going to go to the last one, which was just not that long ago. It's carotenoids in infants. And clearly, we already had the talk today discussing what is, at least getting some information on how to approach the question of what is the proper dose in infant formulas and vitamins. We still are just getting beginning to get at the question of what can we definitively demonstrate beneficial functional and structural effects of these supplementations that we're putting into infant formulas, into prenatal vitamins, all of that. That clearly is our homework for 3 years from now. I know these things take time. And then the last one that I'm going to bring up, this is something that I've done a little bit of work on in terms of carotenoids and the effects of the maternal status, maternal nutritional status, and how it affects the baby's macular pigment at birth, where we published about 2 years ago that maternal zeaxanthin appears to be the driver for the macular pigment levels at birth. Now, is that important? We need to figure out, but clearly, lutein we talked a lot about today, but zeaxanthin does need to be considered. And we've talked a lot about infant formulas. I really am interested in what's, we give prenatal vitamins that have carotenoids. What are those the right things to be giving? How does that affect the mother and the infant during pregnancy? So that's among my charge, and I'm gratified that some of the various sponsors here are at least think that's an interesting question too. So that means we can address some of this. Yes.
I think this is a much higher level question in the context that the consequences may be much more significant, because let's think to the beta carotene model. Vitamin A, which comes from beta carotene, is a transcriptional factor, and that transcriptional factor controls the on-off regulation of multiple genes during embryonic development. And I don't know that we know the answer as to whether any of the apocarotenols or the hydroxyretinols mimic to any extent the role of vitamin A or perhaps inhibit its role. And I think we should move very cautiously in providing carotenoids that might have influence on genetic expression. And so I think it's possibly a wonderful opportunity on the one hand to make the infant ready for the world, but also one we should be thoughtful of, because we really don't know the whole physiology of these carotenoids. Are they involved with transcription and expression?
But on the other hand, we're at least in the United States, nutritionally, these are already on the market, so we need to answer some questions out there.
Yeah, I think a striking image from today's presentations for me was the data Randy showed from how the brain develops in the first 6 months. And as a new parent, I was just blown away by that. I mean, that tells you a lot. So how or should we influence that is definitely something that we should all try and work towards. And it'd be something already I would see will hit the agenda for the next conference, how we can answer those questions, but doing so cautiously, as John suggests.
Yeah, so as you can imagine, as an infant formula company with these compounds in our formula, we are intensely interested in the questions just raised, and I hope we'll be in a position to present useful data. Another gap which I neglected to mention during my presentation in the infant world is that while we are using the lutein and zeaxanthin levels in plasma as a marker, I would love to see continued movement in the direction that Paul is driving, and that is to use the macular pigment as a marker in the infant. If we can evolve that methodology, I think it would accelerate research in this field tremendously.
Yeah, I mean, the RetCam works quite well, but we have about a 6 month up to 6 month window before the kids start fighting back, and then you have to, then you have to put them under anesthesia, and that's not going to go. It's not going to fly in a clinical trial putting kids under anesthesia just to check their macular pigment, or else you're going to have to wait until you can do a spectralis or a flicker photometry at age 8 or whatever that is. Yeah. Yes. Any other comments?
I think we're good. Just to bring up one last point before, I know Lisa wants to have a word before I close the conference. Do you want to go first?
Well, I have one related Yeah, so I just got an email yesterday I have to respond to today. The Chinese want more guidance in where they're going to go in biofortification. And one of the questions that I've had from being at this meeting is, if you have low, we've been breeding for beta carotene, but now we have the genetics that we can breed for beta carotene and lutein and zeaxanthin. It's not going to be the primary compound bred for. But what is like a minimal level to have? In Africa, for example, they eat corn dishes 3 meals a day. That's all they eat. So if you were to be feeding children, but for years, all the time, what are the kind of levels we should try to get to for lutein and zeaxanthin? We're not going to get anywhere near these levels that you do doses and whatnot, but you have the advantage that your study is over 10 years or 20 years. So I would just like some feedback, because I'm going to put that into my thing for China, that we should be thinking about these other compounds, and I just want to be some guidance on some of the breeding we want to do.
Liz probably can. Will you comment on that, Liz?
Well, the epidemiology as far as relationship to AMD risk is around 6 milligrams, and AREDS 2 used 10 lutein 2 zeaxanthin, which was found to be of benefit to with those with low lutein zeaxanthin intake. But I think the reason the Chinese are interested is because, unlike the U.S., they have a dietary recommendation for lutein. And it's quite high. I think it's 10, what is it, 12? Yeah, it's like 12 milligrams a day, which is pretty high, but it's dietary. So I'm wondering if that's their interest in wanting your response to that.
No, I was unclear. My point is not so much, it didn't come from that source. It's like, what is an amount that we can put in of lutein and zeaxanthin we can put into corn, but also there's other crops that might have an effect that, you just said 6 milligrams or 12 milligrams, but the studies are done with these amounts for how long? 2 months, 6 months, 12 months? Are any of them done for 10 or 20 years?
Number of hands, okay.
Well, you can look at epidemiology, and if you look at people following, the current dietary recommendations in the U.S., for example, they're going to get about 6 milligrams of lutein and zeaxanthin. And so you can look to the Chinese recommendations, and they reflect the long term. Clinical trials don't reflect the long term, so you can see whether there's, both health benefits and risks. But I think when you're looking at food sources, I think there's so many biologic mechanisms to moderate the bioavailability that I'm not sure there's the risk that you're talking about.
We're going to have just one or two more questions because it is getting late.
Yeah. Just, Torbert, if you can think back maybe 10 years ago, we wrote a grant together. And it was using your high lutein and zeaxanthin corn and doing the math by just incorporating corn with higher levels, we could really close that gap as far as what people are eating and where we should think they should be.
I'm just reminded that the average consumption out there, regardless of guidelines, and I thought that's where you were going to go with your epidemiology. The average consumption is what, 1.2 milligrams in the U.S. Is it still around there? Just under 2. Okay, but and there's an average macular pigment that goes with it. So I think maybe that's the way to do, is to look at these averages of very big number of populations that give some guidance as to what it is. I think the hidden problem with that is that Paul's first slide. Measurement of macular pigment. We have all these machines, and they still don't measure it all the same way.
And also, you give one subject a supplement who has 0.5 milligrams, or sorry, 0.5 optical densities, and they don't respond.
Just one last.
Can I just comment that China is a huge country. I've only worked in one little bit of it, but there was an area where there was a lot of yellow corn consumed. The lutein levels in those people were off our scale as we'd normally look at them, well over 1 micromole, whereas normally in the UK we would get 0.2 micromoles. So it depends on where you are in China. Some of them have got extremely high lutein intakes already.
Okay, so
okay, we're done.
I'm just going to fit with one last slide. With homework, there always are due dates, as you remember from school. But luckily, you get to pick your due dates here. So just to remind you, about a year from now in Tuscany, Italy, there will be the Gordon Conference. 2 years from now, Lucerne, Switzerland, the International Carotenoid Symposium. And 3 years from now, I'm not sure what the name will be, but don't drop eye from it completely. So brain and eye, okay?
We'll inform you. So just to sum up, I think for anyone that's staying tonight, you want to make an announcement where you're?
Yeah.
For anyone that's going to be in Cambridge tonight.
Who's going to be here this evening? We're meeting at DeLucas at 5:30. You go out by the Porter's Gate and you go to the right, and it's about a 4-minute walk. And we're meeting at the top floor. There's a lounge there. But how many of you are interested in going? So I can tell them. I can give them a number. I've got 1, 2, 3, 4.
5.
5.
You have a party.
We have a party, but we don't need a room. Okay.
So just to, Lisa, maybe can we make your point by email correspondence, or if you want to do it quick, and then because I need to close. Yeah.
Points real fast. If you've done a supplementation study, the backstory is that a couple of years ago at the carotenoids meeting, we had tossed around the idea of a consortium of getting together and actually sharing data. Last year at ICS, we talked about it more. So I went ahead and took the first steps of making a composite table of every supplementation study up to 2015 that has been done in this area. What we'd like to do ideally is a meta-analysis to try to get a better handle on things like low and non-response.
Yeah.
So stay tuned for an email from me with an invitation to participate. And I think using the ICS platform as a means of sharing data and doing those things is probably a very, very good idea. And hopefully within the next little while, we'll have a nice meta-analysis, and maybe you could present that at one of these meetings to look specifically at low and non-response and factors that might contribute.
Thank you very much. No, it's something I think we should do. Just a final comment. Thank you to Paul again for putting in the effort for summing up for us, because it does give us information to go away and think about and work on, and I personally will be doing that with interest. And just a final comment of thank you to everyone for making this possible again. I wish you all a very safe journey home, and I'll correspond via peer review. Take care.